My name is Victor Tybulewicz. I work at the Francis Crick Institute in London, which is a newly formed institute. It's just a merger of two existing institutes, and in a few months we actually moved to a very new building into what is going to be Europe's biggest, or one of its biggest, biomedical research institutes, all on one site, 1,500 people. So my research interest is in understanding the basic mechanisms of Down syndrome, and the approach that we take is to use mouse genetics. So we don't, by and large, deal with the human side of things. Our attempt, we are trying to use mouse genetics in order to understand how the syndrome arises from the extra copies of the genes that are present on human chromosome 21. And in particular, we've constructed a whole series of genetically engineered mouse strains containing larger or shorter numbers of genes, and have used this to map, to identify the regions of the chromosome that contain genes that cause specific phenotypes. And so one of the things that I'm going to describe this evening is the use of such a panel to narrow down the region that contains genes that cause heart defects. So people with Down syndrome have an increased chance of getting congenital heart defects, and around 40% or so have these defects. Our mice also show these, and we've managed to narrow down the area that contains genes that cause this to around 39 genes, within which we already know there must be at least 2 genes. So it's a complex process, possibly more than 2. And we're using the same approach to try and map and identify the genes that cause the learning and memory deficits, locomotor dysfunctions, neurodegenerative processes.